and we have more genomic closeness with the mouse genome. Because of that, we can use mouse as a model in the lab. So all these are the basic research. We got to know about the disease mechanism of various diseases. We have got to know the genes involved in different diseases that can be used as a biomarkers, that can be used to treat the disease by making this drug, through this drug discovery process. So all these things are based on the bioinformatics only, but I have only highlighted a few ones that where we have achieved some success. So next, third part of my talk is how we are applying this bioinformatics in our field of research. So as I told, we are working on vectors. So first project, we are trying to identify the mosquito growth inhibitor. So here we are working on mosquito species that is Aedes aegypti. That is the vector that transmits dengue, chikungunya, and Zika and all that. So as you people already know, mosquito has 4 stages: egg, larva, pupa, and adult. So what we did the RNA sequencing data analysis from 42 different developmental stages of this particular species, right from the eggs to the adult. So then this RNA sequencing data here, I'm showing this heat map where the green color, you are able to see that the green color is for the upregulated genes and the black color is downregulated genes. And most of the genes, if you see that are green color, those are in the early embryonic stages, are in the ovary of the female. So that means they are mostly involved in the embryogenesis. It's actually about the particular class of the protein, not all the genes in the mosquitoes. We are talking only about the RNA binding proteins that are already known to be involved in the embryogenesis. So this is what we are getting through the RNA sequencing analysis also. So most of the genes that are involved in the early embryogenesis. So then we selected around 20 to 25 genes that are upregulated in the embryonic stages and tried to find out, confirm their expression, state-specific expression in the lab using quantitative PCR. So here around 5 genes we found that were clearly showing the expression, mode expression at the embryonic stages, and that we have selected those 5 genes, and interestingly one of that gene is already functionally characterized in Drosophila where it is causing egg mortality. So we are also expecting the same kind of thing in the mosquitoes. Now we are doing this RNA interference to find out the functional implication of these genes on the mosquito growth and development. If we will be able to find out the gene that is affecting the mosquito growth, that we will target for developing the mosquito growth inhibitor. So in the similar line, we are working on one of the gene that is actually involved in making the egg shell around this Aedes mosquito eggs. Because probably people already know that Aedes eggs has the ability to survive any conditions for a couple of months or even sometimes years also. That is not available in the other species of the mosquito. Only Aedes eggs can do that, and for that this egg shell around the shell around those eggs is responsible for that. So some other group in America, they have identified the genes involved in that development of the egg. So we have selected one of the genes that is very important for that egg shell development. That is egg shell development factor, UF1. So we are doing this in silico combinational study to find out the inhibitor for that. We did first homology modeling to find out the structure of 3D structure of that gene. So we found 5 such models. We did their validation by Ramachandran plot, and then some other parameters by molecular dynamic studies. Then out of this, one was showing good stability and good structures that we have selected, and then we docked it with the library of chemicals to find out the inhibitor molecules. 5 of the compounds we have identified that is showing the inhibition, and then we did the find out the residues, which particular residues are actually interacting with those compounds, because we have to see which part of the protein these residues are present, whether they are stable or not. And then we are doing the molecular simulations for finding out whether this molecule and the protein pair is stable or not. So in that way, we are trying to find out the inhibitor molecule against this particular gene that is very important for the actual development of mosquitoes. That is one of our project right now. And the second project that is funded by SCRT is a startup grant, where we did molecular ecology of this particular species, Aedes aegypti. So here I am sharing the Madurai data, that you can see the satellite map that is for the Madurai city, where we collected these mosquito samples from different locations within the city, as well as different outskirts areas, and then we did this population genetics analysis, and what we observed that the mosquito populations that are present within the main city area that are genetically similar to each other. But the outskirts area like Pashchimalai, Tripurapuram and Avaniyapuram, they are genetically distinct from the populations that are present within the area. That shows the connectivity between the areas, because people already know those areas are not that much connected with the main city area, so that might be the reason, because of the genetic connectivity between the mosquito populations too. So that same thing we are doing across the country through these climatic zones. So far we have analyzed around 24 populations all over India, and we have seen the pattern associated with the geographical regions. So this data is we are still analyzing, and maybe that will yield some more observations based on that. So this is our second project that is involving this bioinformatics analysis and data analysis. So third project, we are working on Bacteria-Vector, that is the pathogen that causes lymphatic filariasis. Here what we did, we are doing this in silico analysis, particularly to identify the genes that can be the candidates for the drug development and can be used as a biomarkers. We analyzed the whole proteome of this species. Then we found out the genes that are not homologous to the humans, because that we cannot target, that are same for the humans. So those non-homologous, human homologous genes that will be considered as the pathogen specific. Then we try to find out the essential genes that are very essential for the pathogen using the essential gene database that is already present publicly. Then we try to find out their subcellular localization of those genes, whether they are present in the cytoplasm or they are extracellularly excreted, because those are present in the cytoplasm, they can be targeted as a drug, because that will be present within the pathogen, so they will be the drug targets. But those that are those are secreted outside, they can be either used as a biomarkers or can be used as a vaccine targets. So then for the drug targets, we are doing the modeling and the docking, and for those candidate biomarkers, we are finding out their antigenic properties and also trying to find out whether they are species specific, so that species specific only can be used as a diagnostic marker. So this is the in silico work and computational work that we are doing to identify the biomarkers for this particular pathogen species. So besides that, we have recently received funds from ICMR for doing the genomics and stage-specific transcriptomics of the same, this Bacteria-Vector, to identify the biomarkers and the genomic diversity. So that has not been started yet. We are just doing the sample collection and all. So these are the two publications, and some of them are not published yet, but submitted. So this is our small group who is working with me since last 4 years. So this is all about our today's topic. I don't know whether it's finished. So any doubts?